In this lesson, we'll be focusing on interactions between microbes and humans. So, oftentimes in this class, we'll obviously be focusing on infectious diseases. There are many different types of diseases, though. A disease, technically, is anything that causes someone to deviate from an otherwise healthy norm. So, while diseases can be caused by infectious pathogens, and that's what we tend to focus on in this course, diseases can also be a result of diet, genetics, and even aging. So in this class exclusively, we're going to be focusing on infectious diseases, which an infectious disease is a disruption to one's body, such as cells and tissues and organs, as a result of microbes directly, for example, a virus infecting a cell and then destroying that cell, or products that microbes produce. So for example, a bacterium that produces a toxin that affects our body in some way. And infectious diseases do run through a continuum, so it typically starts with contact. So we come in contact with an infectious pathogen, and then it enters our body. Once it enters our body and starts to grow, at that point, it would become an infection. And then once it's grown to the point where it starts causing damage and signs and symptoms are present, then it becomes a disease. Fortunately, not every contact is going to lead to an infection, and not every infection is going to progress to a disease. If every contact led to an infection, we would never be healthy. We would be sick all the time. But we come in contact with pathogens all the time, and we have immune defenses, innate immune defenses, that prevent the bacterium or virus or whatever it be from getting into the body and establishing an infection and growing. So for example, if you breathe in something potentially pathogenic in the air, you have nasal hairs that could potentially block that pathogen from getting deeper into your respiratory system and causing an infection. Even if it does get in, it doesn't necessarily mean that it's going to progress to a disease where you have signs or symptoms. Because again, even once it gets in, we have immune defenses such as phagocytic cells that can engulf and destroy a pathogen before it becomes problematic. But for the sake of this course, we are going to progress through these different stages of contact, infection, and disease. So before we get to the problematic microorganisms, the pathogens, we get to talk about the good ones. So the good ones are called normal biota, or you may have heard them being called normal flora. So these are the microorganisms that are found in or on our body and do not cause us any harm. A lot of times they can actually benefit us. They will typically be present in either a commensalistic or a mutualistic relationship with their host. So by that I mean, in a commensalistic relationship, they would benefit from the association with us. However, we would be neutral. We wouldn't be harmed, but we wouldn't be benefited either. In a mutualistic relationship, both the host and the microorganism would receive some type of benefit from that relationship. As far as where microorganisms are found, generally they're found in places throughout your body that are exposed to the external environment or have an opening to the external environment. So for example, your skin, fully exposed to the external environment, so you have a ton of normal biota there. Your digestive system is exposed both at the beginning and end to the external environment. It has openings, so that you'd have normal biota throughout. Upper respiratory system, you have normal biota throughout there. You don't have normal biota in areas of your body that are completely closed off, such as internal organs, the blood, nervous system, those types of things. But if it's a portion of your body that is exposed to the environment or has an opening to the external environment, you likely will have a ton of microorganisms, beneficial microorganisms, or normal biota as we call them in those areas. So normal biota do quite a few beneficial things. I mean, they help stimulate our immunity, they produce vitamins for us, they help with digestion. We could do a whole class on all the benefit that normal biota have within the body. But one of the very much beneficial aspects of normal biota is this concept known as microbial antagonism. So microbial antagonism is how essentially the good microorganisms help protect us from bad microorganisms. So it's the antagonistic effect that good microorganisms, or the normal biota, have on pathogenic microorganisms, which are the bad ones. So we kind of just think of this as how normal biota protect us from getting infected with pathogens. So there are 3 different mechanisms that normal biota can use to engage in microbial antagonism. So one has to do with taking up attachment points. So we have billions of normal biota in our gastrointestinal system alone, and in order to stay in our gastrointestinal system and not get expelled through something like peristalsis, which moves intestinal contents throughout the gastrointestinal system, they have to adhere to cells and tissues and receptor sites, typically with their fimbriae. So if the normal biota are taking up all the attachment points and a bad pathogen gets in, it's not going to have anywhere to bind. And if it doesn't have anywhere to bind, then it's eventually going to get expelled out of our body. You know, peristalsis can move it down to the stomach where it would get killed by stomach acid. If we're talking about respiratory, we could cough it out, we could sneeze it out. So from a pathogen's perspective, it wants to get in and to attach to something so that it can stay in the host. And if the normal biota are taking up all the attachment points, it makes that very difficult for the pathogen. Another thing that can happen is the normal biota can alter the environment in the host to make it unbearable for pathogens to the point where they couldn't potentially grow in those areas. One of the most common things that's done is the normal biota can make the environment acidic so that pathogens are not able to grow in that location. Most pathogens prefer a neutral environment, not all. So if the normal biota are present and they're making the environment more acidic, it's problematic for a pathogen. A really good example of this is Staphylococcus epidermidis on the skin. Staph epidermidis feed off of our skin oils. They're a healthy normal biota, but they feed off of our skin oils and they release acids. And that gives our skin an overall acidic pH, so that if something potentially harmful, such as a pathogen, lands on the skin, it generally wouldn't be able to survive very well. One, because of the acidic environment that the normal biota have created, but then also because of the high concentration of salt due to sweat. So many microorganisms can alter the environment by simply making it an acidic environment so that pathogens cannot grow in that area. Another thing is quite simple. Normal biota can just use up all the nutrients. Microorganisms need food in order to obtain energy, to grow and divide. So if the normal biota are present in a specific area and they're using up all the nutrient source, then there's nothing left over for the pathogens to use as an energy source, and they essentially die off. So these are just the various ways that normal biota can serve to protect us from pathogens. So one thing that we need to understand is when we first get normal biota. At this point in your life, you have cultivated your normal biota, you have trillions of healthy, beneficial microorganisms throughout your body. But when do we first initially get normal biota? So for years and years and years, we believed in something called the sterile womb theory, which was a belief that in utero, that was a completely sterile environment, and that a baby did not get colonized with normal biota until the birthing process. So as long as they were within the uterus, it was completely sterile, they had no normal biota, and then once they've gone through the birthing process, that's when they would first get normal biota. That has actually been disproved, and they've actually looked at bowel movements, fecal movements from babies, and they've seen that their first stool actually has normal biota in it. So that tells us there must be normal biota present in utero. So now it is known that at some point in utero, fetuses do get normal biota, typically present within their gastrointestinal systems. So that's when they first initially get it in utero, and then they definitely build on it extensively during the birthing process. Now, there has been research that has shown the mode of delivery can affect how much normal biota they get. So it has been shown that a pregnant woman will have significantly more healthy bacteria in the vagina than a non-pregnant woman. And the idea behind that is that healthy bacteria like lactobacilli that's present in the vagina is going to be passed to the baby during vaginal delivery. So during vaginal delivery, the baby will get all this like healthy lactobacilli, bifidobacteria, which is just a beneficial bacteria that helps with digestion, different types of staphylococcus, streptococcus, all healthy normal biota. Obviously, not all babies are born through vaginal delivery, though, and there has been research that has shown babies born through C-section do have diminished normal biota that can make them more susceptible to autoimmune disorders, other infectious diseases, problems with digestion. So it is a concern. So obviously there needs to be a way to kind of supplement that normal biota in babies that are born through C-sections. Now, breastfeeding is something that can definitely help. Breastfeeding allows them to get normal biota, especially bifidobacterium and lactobacilli that are really beneficial. So breastfeeding can often be used to kind of supplement the normal biota. But one interesting kind of thing that's being used, that's being researched, is this idea of what they're calling vaginal seeding, or even sometimes it's called a microbial bath. And what they do is during labor, prior to doing the C-section, they put this gauze wrap in the mother's vagina to soak up additional healthy bacteria like the lactobacilli in the vagina. And then when the baby's born, they wrap the baby in that gauze wrap. And that applies all the normal biota from the vaginal canal to the baby. And they've tracked these babies throughout 6 months post-delivery, and they've seen that they have the same amount of normal biota as babies that were born through vaginal delivery, and they're no longer prone to some of those complications that we talked about. So this may make sense to do routinely in the future with all babies that are born through C-section. They're still kind of researching and learning more about that, but it has shown to be promising. Nutrition can influence normal biota as well. Obviously, no matter how the babies feed, they're going to get normal biota. So if they're fed through breast milk, then obviously they would get normal biota present in the breast milk from the mother. If they are fed through formula, there are probiotics and prebiotics that are present in the formula. Research has shown that breast milk is the most beneficial. I mean, both are beneficial, whether you choose to bottle feed or do formula. In both situations, you are going to get healthy bacteria that will colonize within the gastrointestinal system. It's just that breast milk has a higher concentration of this bacteria called bifidobacterium, which is just a great bacteria. It helps with inflammation. It helps maintain the acidic pH in the stomach. It helps with digestion. So just getting more of that is really beneficial, and how to get the most of that is through breast milk.